Okay, so today I'm going to talk to you about some of my research. I do research with horsehair worms in the phylum Nematomorpha, and we do work in the lab, on the field, and recently, as of like maybe a week ago, we're going to do some internet research. Um, so to kind of go over the life cycle, if you study parasites, life cycles are critical to understanding the parasite that you're studying. And this particular parasite infects insects, so things like crickets, roaches, mantids, and it manipulates that insect into entering the water. And once it's in the water, the adult worms will emerge. And you can see in this video, the worms emerging from this cricket that was dunked in some water. And those worms will, there's male and females, they'll mate, they'll lay eggs. The eggs develop into larvae, and then the larvae will insist in another host. So typically an aquatic insect, such as maybe a midge or the mosquito larvae, or perhaps mayfly larvae. They'll insist in there. And then when that mayfly or mosquito becomes an adult, so then when the mosquito becomes an adult, flies around on land, or this midge flies around on land, then the cricket will eat it and become infected. The cyst stage will also appear. They'll also insist in snails, aquatic snails, but we don't think aquatic snails like leave the water and are eaten by insects. But I will return to that idea later. But right now we'll just call the aquatic snail kind of a dead end host. The reason I have the aquatic snail in this picture is because they're actually kind of important for my research. Finding these adult stages in an environment, in a particular creek at any particular time is kind of difficult. The worms, they just look like little hairs or vegetation, so they're hard to find. And they're also very seasonal. So if I go out in July to one particular creek, I can find them in July, but any other time of the year, I can't find them. So the cyst stage, however, they're always in the snail year round, and the snails are always there year round. So we can collect those snails and find the cyst stage in the snail and know that the horsehair worm's in that particular creek. I can also take those snails and put them in the -80 freezer. And then like in February, when it's really cold in Omaha and I don't want to go out in the field, I can take those snails out of the freezer, thaw them, and feed them to crickets and infect the crickets. So I can maintain the life cycle of the horsehair worm in my lab. So what I thought we'd do today is kind of go through some of my work that I've done in the lab, some work I do out in the field, and more recent kind of stuff on the internet. So when these adult worms are out and they emerge from those insects, they have to find each other. They typically, not always, but typically the male and female worms, depending on the species, and they have to find each other in these creeks, and the creeks are kind of moving pretty fast. And so the idea is, are they encountering each other randomly, or is there, are they using maybe a pheromone to attract each other? When they do encounter, the male will kind of wrap around the female. So we call this kind of wrapping behavior, and then deposit sperm on the female. And so to kind of test this idea, what we did, we had a little aquarium, we filled it up with some water, and we said, well, if I took a male worm and a female worm and I put them in the same tank, they should encounter each other much faster, much sooner than if I put a male worm in and another male worm, or a female worm and another female. So we ran 68 15-minute trials on recently emerged adult worms. So again, had male-female pairings, but also male-male and female-female, and measured the time to first contact. And then we also determined, did they do that kind of did the males do that wrapping behavior? So in 19 trials, the worms failed to make first contact. 14 of those trials were from male-female pairings, 5 from female-female pairings, and in 6 trials, both worms were active in the tank. So I analyzed the 49 trials in which first contact was made. And so in female-male pairings, 64% of those resulted in contact. In male-male pairings, 100% result in first contact. And in female-female, 64%. And then you can also see 64% of the male-female pairings resulted in wrapping behavior. Females do not undergo that wrapping behavior, so nothing happened in those trials. If you look at the time to first contact, we can see that, yeah, female-male pairings took less time. So it's consistent with the hypothesis. It took less time than same-sex pairings. And that was, looking at the statistics of that, wasn't significant, 0.23. Again, you can look at it when you kind of separate out the male. So this one was just same-sex, male-male and female-female, but you separate it out and do an ANOVA. Same sort of result, wasn't significant. But the timing is consistent with the hypothesis. So in this case, you can see I did it in 5 centimeters of water. So well, what if we kind of reduce the amount of variation, because maybe that pheromone's getting diffuse in the aquarium tank, and so if we had more of a 2-dimensional system and they're working in, and just have 1 centimeter of water. And similar percentages, 92% with female-female making that first contact, and 100% of them having that wrapping behavior. So, if you compare 5 centimeters versus 1 centimeter, the numbers, are more consistent, I guess, with the hypothesis that male and females will encounter each other in the tank. And then certainly with the timing, female males, encountered each other much quicker than same-sex pairings, and that was significant at the 0.02 level. And even if you kind of separate it out and do the ANOVA, 0.06, so almost significant. So when you reduce the variation and you just had that 1 centimeter of water, it does yield results that are more consistent with the hypothesis. But then it also raises the question, like, if you've got this, if you're in a creek, right, how do they, that's much more variation than in this 10-gallon tank. But we did have enough evidence to kind of justify further experiments. So we want to kind of record those trials and maybe do an analysis of their movement. And then maybe if we have access to an artificial stream, we can also conduct similar trials there. We're also going to look to see at mating preference. So we'll have a worm kind of in the middle of the aquarium, and then, the wrong way, and then place stimulus worms at each end, and just see, okay, kind of lift up the glass and see where does the that focal worm in the middle, does it prefer one worm over the other? So we can kind of look at mating preference, and different types of things along those lines. One thing we think we isolated, a sex pheromone. We did some chemical analysis of the cuticle on the worm and the water that the worm was soaked in, and isolated a chemical compound that we think might be serving as a pheromone. So we just got that a week or so ago. So we're going to do some trials in the lab to see how these worms respond to that chemical. Okay, so I will be returning to this little picture of the life cycle numerous times, so you can, don't worry if you didn't get it the first time. But I kind of mentioned that the snail was a dead-end host, or we think it's a dead-end host, because crickets, the aquatic snails have to be in the water. They don't turn into flying insects or like the paratenic or the aquatic insect host does. So, but there was some anecdotal evidence. A colleague of mine, when he was doing these studies, he would go out in the field, he'd collect snails, put them in a bucket, and then reach in the bucket and pull out snails and dissect them to see how many cysts that they had in them. And he thinks that, when he was pulling snails out from near the top of the bucket, those snails seemed to have like the most cysts. And when he had reached down further in the bucket, the snails at the bottom didn't seem to have that many cysts. And so when you go out in the field, you can see like at the edge of the creek, like some snails are like kind of stuck out above the water line. Some are at the water line, and then some are below the water line. And we also observe crickets. If there's a snail at the edge that's kind of dried up and dead, we've seen crickets, grasshoppers kind of eating those dead snails. And so if these cyst-infected snails, if they do spend more time at or above the water line, then they're going to be more susceptible to drying out and being consumed by a cricket host. I had a couple of students working on this. We kind of scored like their behavior, observing the snails, and this is based on a previous paper. I'm not going to actually spend too much time on the scoring because we actually didn't end up using it. But snails do exhibit a limited amount of behavior that you can kind of score. But what we focused on, so what we did was we had mason jars, we filled it up with water. We had snails that from a site where we knew horsehair worms were present. We had snails from a site where we've never seen horsehair worms. And we basically put one snail in each of these jars. And then the snails were allowed to sort of climatize overnight. And then in the morning, we got up early and measured their position and scored their behavior every 3 hours. And we didn't really find a significant difference between the cyst-infected and uninfected snails. There was some evidence that maybe infected snails moved more. But, as you might imagine, we only had 3 snails. And then my students weren't able to collect data during one part of the trial. So this is something that we kind of want to return to. But the implications of it is, obviously we're going to have to repeat it with a larger sample size, and maybe record, like video record the snail movement. But we, because we were measuring their behavior over time, over a period from like 6 to midnight, we think that maybe we should focus more on kind of diurnal time differences. So maybe snail behavior probably fluctuates just naturally over the course of a day, but maybe the ones that are infected with cysts, if during the middle of the day when the water levels are most likely to kind of recede in the creek or pond that they're in, maybe that's when they're more active, or they're more likely to exit the water. And then that would also correspond to a time, when crickets are sort of more active at night, and so you have these kind of dried up, or these snails are kind of stuck stuck out of the water, and now crickets are just becoming more active and foraging. So we're going to take a look at kind of diurnal time differences. Okay, so kind of moving now into looking at field work. And here we'll talk about Paragodes varius. So Paragodes varius is a hairworm species that's found in Nebraska, and it's fairly well studied. We know that the cricket is a terrestrial host for it, and the little GIF, the little video I showed you of the cricket, of the worm coming out of the cricket, that was Paragodes varius. So we have sites in Nebraska where we know they're present, and again, like certain times of the year, you can find the adult worm. And in September, we were able to find the adult worm in this particular site, and we're able to find the worms will like knot together. And this is Another word, another term to describe nematomorpha are the Gordian worms. So the Gordian knot of Greek mythology comes from the fact that many of these worms will, or some of the species will kind of tangle themselves into these knots. And we don't know what the knotting behavior, is for. Like, we don't know what it actually, how the worm benefits, if at all, from this knotting behavior. Some studies have suggested water regulation. So maybe if these worms are fairly small, susceptible to drying out, and maybe if they're in these knots, it better helps them regulate water. Some have suggested maybe mate facilitation, right? You enter into these knots and you find a mate. Or predator avoidance, right? If you're in a large knot, it's less likely to be eaten maybe by a fish or something with a limited mouth gape. When we go out in the field, we only find males in knots, and that's consistent with other studies. And so we decided, well, we're going to go out in the field, and we will go during a time of year where we know the adult worms are out there, and we'll look and see, what's going on with these knots. And we hypothesized that adults occurring singly and adults occurring in knots, they're going to differ in terms of their weight and length, right? So if this is about water regulation or predator avoidance, maybe smaller worms, for example, might gather into knots, and the larger worms might, go on their own. Or mate facilitation, again, usually larger worms. The larger the worm, the more gametes it has, and so maybe the larger worms might be found in knots to facilitate to finding a better mate. But we just figured they're going to differ somehow in terms of length and weight. So we constructed a transect in a creek, and we just moved along that transect and collected all the adult Paragodes varus that we could find, and noted, like, were they in these knots, or did they occur singly, just alone? And we also noted if there were male or females, and then were they occurring on vegetation, like in this one, or were they sort of in the mud? And then we brought them into the lab and took the dry weight and measured the length of all the worms. So we found that the adult worms, they occurred 51 times. And so we had 96 hairworm occurrences. And all of the worms we found were males. They were associated with vegetation, so not in mud. 36 of them occurred singly, and 15 occurred in knots. And so there was a significant difference in terms of the way they occurred, in terms of knots versus singly. The knots range in size from 2 to 8 males. Average was 4 males per knot. Hairworms that occurred singly and did not differ in length, dry weight or length per dry weight, compared to worms that occurred in knots. So there was no difference in terms of length and dry weight with the worms in knots versus singly. Why is this not? Oh, I see. Hold on. Okay. So implications of this. So we know size, again, is going to correlate with fecundity. The larger the worm you are, the more eggs you can produce. It corresponds with a tendency to dry out. Smaller worms are going to be more susceptible to drying out, and also their ability to escape predation. So this lack of size difference suggests that knots are not playing a role in any of those functions. We do think that maybe worms exhibit what's called stigmataxis. So this is just this tendency for, if you ever, mayflies, for example, will do this. They will just cling to things. It doesn't matter what it is. They will cling to each other. They will cling to twigs and branches and leaves. They just cling to each other. So stigmataxis is just this tendency to sort of cling to things. And maybe worms just do that. And then the knots are just forming when the water currents just sort of randomly push the worms together. So maybe there really isn't any sort of sex pheromone or aggregation pheromone. But again, this is something that we can look at in the lab if we can sort of isolate those pheromones. So that work was published relatively recently. This work hopefully will be published this year. It was accepted, but this is with another species of horsehair worm, Cordurus morgani, also found in Nebraska. And unlike Paragordes varius, we don't know a lot about it. So we don't know the terrestrial host, we don't know the aquatic host. But we can go to this creek near Lincoln, Nebraska every July and find adults. And the adults, unlike Paragordes varius, that clump into these knots, the adults here will wrap themselves around branches and twigs, and they will lay their eggs. You can see this one laying eggs here. And the eggs are kind of have a very distinct pattern to them. So, but we only find them in that one particular creek. And, why is it, are they only in Elk Creek and not kind of in neighboring creeks? And then what is the terrestrial host, right? So we wanted to ask that question. And this kind of fits in the kind of an overarching goal that I have for all my research, and the idea of using these hair worms as indicators of ecosystem health. So, I've shown you that the life cycle of these hair worms, and you can see that they have multiple hosts. And if any of those hosts are not present in the environment, then the worm will also not be present in the environment. So if you have a diverse assemblage of these particular parasites of these horsehair worms, and there's at least 3 species in Nebraska. But if you have a diverse assemblage of those horsehair worms, then it should indicate kind of this healthy functioning ecosystem, because all of their respective hosts also have to be present in the ecosystem. So we hypothesize that the terrestrial host restricts the presence of Cordurus morgani to Elk Creek, and as opposed to the neighboring creeks. So to test this, we sampled Elk Creek and nearby creeks, and Elk Creek flows under 2 bridges. And in the United States, you can get a collecting permit, at least in Nebraska in the United States, you can get a collecting permit to collect, under the bridges, because those are considered public property, and you don't have to worry too much about sampling things on private property. So you have to, even though we did talk to the neighbors and they were okay with us sampling a little further along the creek, but because Elk Creek flowed under these 2 bridges, we could actually sample under both bridges, and then actually really along the entire, almost this whole portion of the creek. So we were able to kind of extensively sample this in July of 2017. Also looking at chemical physical parameters. So we have a probe that will measure pH and nitrate levels, temperature, dissolved oxygen. So you can see just the sites that we went to. The Elk Creek site is down here in the corner. And you see the 2 bridges there. But then we went to another site we called Maple Creek, and it was about 4 miles away. And we actually did find horsehair worms at that site. And then we went up to this creek, which we called Upper Elk Creek, because it's the same creek as Elk Creek, it's just upstream. And we found horsehair worms here. And then we went to West Oak Creek, and this site, we've never found horsehair worms there. In 2017, 2018, and 2019, we've never found horsehair or any horsehair worms there. So we're able to sample these 4 sites at 3 different first order streams, and we kind of installed these little chopsticks. Basically, we strung together chopsticks and put them in the creek, hoping that the worms would sort of wrap around the chopsticks. So we had these little stick traps. We had pitfall traps to collect the terrestrial insects. I have this YSA probe that looked at temperature, dissolved oxygen, conductivity, salinity, pH, nitrate. We also physically measured the depth of the stream, the width, and stream flow. The stick traps actually weren't super successful at collecting the adult horsehair worms, but they were pretty good at collecting vegetation that kind of moved through the stream. So we also used the stick traps to collect vegetation. Here you can see, like this is a little popsicle. Actually, this was like a art stick you can buy at like a little craft store. And you can see the adult worm on the popsicle stick here. And then we just strung them together on a fishing wire and tied it to a stake and put it in the creek. So it wasn't highly successful. There's one worm that we got on there, but for the most part, we didn't get a whole lot on those things. Here's an example of some of the pitfall traps, and we just, all along the creek, we would install these pitfall traps. Some of the baited traps we used to kind of, the pitfall traps, the insect just fell into it. Baited traps that were attracted to a bait that consisted of oats, peanut butter, and stale beer. Here we are kind of measuring the various chemical properties of the creeks. Here is how we measured the physical properties. So again, we constructed this transect, 10-meter transect, and measured the width at 0, 5, and 10 meters. Did the same thing with depth, and then flow rate was measured by dropping in kind of this little fishing buoy and measuring the time it took to float 5 meters, and we did that 5 times in the creek. So here are our worm stick traps. Instead of collecting the adult horsehair worm, they're actually very effective at collecting the vegetation. So we scraped off all the vegetation and measured the dry mass of that to sort of estimate that. Okay, so in 2017, we collected 26 adult Creighton margani in Elk Creek, 9 at Upper Elk, 19 at Maple, and there's nothing at West Oak. We also collected some specimens of a third species of horsehair worm, Gordius robustus, that is not one that I'm actively studying right now, but it is present here in Nebraska. Okay, so we did find evidence that the wood cockroach, Parcoblata pennsylvanica, serves as a terrestrial host for Creighton margani. So we had a vial that I was collecting insects, and I saw this little roach running by, and I also saw this beetle running by. So I grabbed it, threw it in the vial, brought it back to the lab, and sort of like forgot about it until the next day when I saw worms had emerged in the vial, and there was a dead roach, and the beetle was still alive. But we're pretty sure it came from the roach, because the roach had like burst open and these worms were flying around, and you could see like a piece of the roach on the worm, and the beetle was fine. So we're pretty sure it came from the roach. And we also have done some experimental infections showing that you can successfully infect the wood roaches with the Creighton margani. And so when we look at kind of putting it all together, the occurrences of the worm and the occurrences of the roaches, we did find wood roaches at Elk Creek, Upper Elk, Maple, but we also found them at West Oak. And West Oak was the only place we did not find the adult horsehair worm, suggesting that our hypothesis that the roaches are probably not restricting the range of Creighton margani, right? Because the roaches are everywhere, but the worm was not at West Oak. So maybe kind of looking at some of the other parameters, pH did differ significantly between West Oak and Upper Elk. So there was a little difference in pH. The flow rate also differed between West Oak and both Elk Creek and Maple, but not with Upper Elk. And vegetation dry weights also differed significantly in West Oak and both Upper Elk and Maple Creek, but not between West Oak and Elk Creek. Okay, so implications. So the terrestrial host does not limit the distribution of Cordilleras Morgani. The persistence of Cordilleras Morgani in Elk Creek. Now I have data that goes back from another colleague of mine who, when he was a graduate student, studied Elk Creek and knew that these guys were there. So they've been in there for at least 16 years. I guess now we're going on 20 years. So they're pretty they're resistant to climate change, agricultural pollutants. It's a very agricultural area in Nebraska. We think I'm not really studying Gordius robustus, but because we find both of them in the same creek, maybe they share similar ecological niche. Also, even though we didn't see it in some of the sites that we sampled, they may be present in other Nebraska creeks. Another colleague of mine found them in a creek also in Nebraska, a little further away. We didn't find like any single physical or chemical factor that correlates with the presence of Cordilleras Morgani. But there's some evidence in the literature that pH may play a role in other species of Cordilleras. And this is the first paper that will say that pH may limit Cordilleras Morgani distribution. Vegetation, we kind of looked at vegetation because we know Cordilleras Morgani likes to wrap around twigs and branches, and so maybe vegetation might play a role, but it doesn't seem to play a role here. Currently, we're looking at atrazine. This summer, we're going to measure atrazine at a bunch of sites and see does atrazine, which is a herbicide farmers use in Nebraska, does that correlate with the presence or absence of Cordilleras Morgani and or maybe some other horsehair worms. And then, of course, the other question we have is if the terrestrial insect doesn't limit the distribution, maybe the aquatic, the parotenic host does. And so we want to know what that is. So more recently, we want to know what the likely parotenic host. So the parotenic host just refers to this kind of technical term, saying that it's transferring the parasite from one environment, the aquatic environment, into the terrestrial environment. So they call it a parotenic host, sometimes a transfer host. And so, we want to know, like, what is the likely parotenic host of Cordilleras Morgani? And I say likely because it's really difficult to figure it out. This was a paper from, I think, 1964. And they went out and they collected a bunch of insects and they dissected them to see if the cyst stage was inside them. And the cyst stage was inside lots of these insects. And it appears that that the larval stage will insist in all kinds of things. So it insists in those aquatic snails. It insists in mayflies and these Corixidae, which are these little water boatman things. Black fly larvae, they'll insist in that. Interestingly enough, the black fly larvae, they found them, but infected black fly larvae could not pupate successfully. They would just die. The uninfected black fly larvae could. So, if they can't pupate and they die, it's probably not black fly larvae, right? Because they're going to just, the cysts will just die inside those larval stages. So there's a lot of suspects out there, a lot of likely suspects. Again, the aquatic insects are hosts, but then you have these snails and trematodes and leeches and fish that they'll also find the cyst stage in them. So to serve as a parotenic host, the host has to transfer that cyst stage from the aquatic environment to the terrestrial environment. So again, trematodes are probably not going to do that. Leeches, probably not. Even the snails, we don't think are playing a big role in that. certainly not fish. But there's this clue, at least for Cordurus morgani, right? Unlike other horsehair worms, Cordurus morgani will deposit its eggs on sticks. And so a likely paratenic host should occur like when the eggs are deposited. So, in the timing is important. The cyst stage has got to be both in kind of the larval stage of the host, but also in the adult form of the host, right? Because with the black fly larvae, it might have been in the larval stage, but not so much in the adults. And then that adult now is going to transfer it into the terrestrial environment. So the terrestrial host has to encounter it, consume it, and become infected with it. So all those things, boxes have to be checked. Now, there are some aquatic insects that we know they feed by kind of scraping algae, detritus from the surfaces of submerged sticks, rocks, and barks. And so you can see this mayfly larvae here is kind of this feeder scraper kind of behavior where they're just sort of scraping the algae off, in this case, a little stick. So scrapers should be more exposed to cysts and could serve as a likely paratenic host. So we hypothesize that these scrapers, if you collected, went out in the creek and collected a bunch of sticks and looked at the sticks and looked to see, do they have eggs or do they not have eggs? So sticks with eggs, sticks without eggs, and then removed all the insects that were on them. The scrapers that were removed from sticks with eggs, they should have a high prevalence of infected invertebrates. They should have a high number of cysts inside them. If you took sticks that didn't have any eggs and you looked at kind of the insects that were on them and looked at the non-scraper insects, the invertebrates, they'll have the lowest prevalence of infected invertebrates, the lowest number of cysts. So that's what we did. Went out, collected these sticks, brought them into the lab, scored them as having eggs or lacking eggs. And it was kind of consistent here. 82% of the invertebrates that found on sticks with eggs, they were infected. Of the scraper invertebrates are infected. And then the intensity was pretty high. It was 17.5 average intensity. We also, though, found scrapers also had high average intensity, when they had sticks with eggs here, or sticks without eggs here for the scrapers. Okay, so I'm not going to go through all of this here, but let's just say that the likely suspect here are these flat-headed mayflies. They belong to the family Heptagenetidae, and you can see the larval stage and the adult stage here. And this was really cool. We went into the lab. I said, well, they're wood roaches. We don't know what they really eat. I just assumed they were called wood roaches because maybe they eat wood. But if you drop a mayfly, and we took the wings off the mayfly, so it didn't fly away from the roach, you drop it into the vial with the roach, these roaches, they will eat those mayflies. And then approximately 62 days later, on average, they became, the worms emerged after that. So they will eat them, and they do successfully infect the wood roaches. So again, this study kind of increases our understanding of the life cycle. It kind of is, if we can identify these paratenic hosts, which is always a little kind of problematic. But in the case of this, like those mayflies, they're flat-headed mayflies, they're relative to other invertebrates in the creek, they're pretty sensitive to pollution and other stressors. And so, again, it plays into this idea that if you have parasites or these complicated life cycles, it could indicate a healthy, functioning ecosystem. Okay, so the final thing I just want to talk about here, and I don't really have a lot on this one, but hairworms and the internet. So this, and this is also kind of an invitation to for collaboration. for anyone who's out there and think, oh, these are really cool, I would love to study this, but I can't for some reason. Well, in fact, you can. So on the internet, there's tons of digital data now, and there was an article that came out this past year called referring to this idea of eye parasitology, and it kind of was a reference to this eye ecology paper that came out, but we're talking about parasites. So, and they define eye parasitology as the study of patterns and processes in parasitology using online data stored digitally, publicly available, and often, but not always, generated for other purposes. Okay, so we're talking things like social media, so like Twitter, Facebook, Instagram, right? We have people are posting all kinds of pictures on there. There are what I would call like citizen science sites. So iNaturalist is the one I use, but there's like Bug Guide, and there's another one that came out, Quest a Game, right? Where people like, they find interesting things and they're a little bit more serious about identifying like, what is this? And they upload it to these sites. And they typically will record like, where it was found, the date they found it. And in some of the cases with the parasites, some host information might, if you're lucky, might be available. So, if you go on iNaturalist, for example, and search for horsehair worms, 972 examples so far. This is just a couple of days ago. There might be more on there, right? And so they'll have like the picture of the worm, some identification, the location, the date that they made the observation. I also was kind of curious, okay, what was going on in India? Only 14 observations. So if you're out there in India and you come across something, it looks like a horsehair worm, upload it to iNaturalist and you can get involved with like as a citizen scientist and upload your observations there. When you click on some of the observations, sometimes they may actually tell you the species. This is my observation, so I knew it was Cordurus morgani. I knew what they had done, so I was able to give the species name. Most of the time, they're not going to give the species name or identify it so accurately, but sometimes the species name is there. I currently have a student. He wanted to do research with me in the summer, but wasn't able to spend the time in Omaha with me. He's from Hawaii. And so he wanted something to do while he was in Hawaii. I'm like, well, okay, go on iNaturalist. And he just sent me the data this morning. Well, I guess last night, whatever it was. I haven't looked at it yet, but he went to the United States and looked at all the observations kind of in California. And then he picked some states on the East Coast that, in terms of the geography, the land area, and also in terms of land, or in terms of population size, matched California. And I don't remember what the states were. I think it was like Virginia, New Hampshire, a couple other states. But those states on the East Coast were equivalent to all of California in terms of land area and in terms of population, because you don't want to bias it. And so he just looked at the observations of Nematomorpha, and we're going to see like, are there more on the West Coast versus East Coast? So you can kind of generate some of these hypotheses and say, okay, well, maybe in the Appalachian, it's the East Coast is the Appalachian mountain range, which is an older mountain range, versus like the West Coast, where you have the Rocky Mountain, and that's kind of a newer range. And so maybe, like, if I had to guess, I would say maybe there'd be more observations on the East Coast. It just seems an older mountain range, an older area, and maybe you're going to find more observations there because you just have more species of horsehair worms. But we don't know. We have no idea. Anxious to look at the data you just sent me. But the iNaturalist, you can also use to identify new sampling sites. So, there are some places in Nebraska. I'm going to go to Iowa later today and like, oh, maybe I'll stop by at this site and see if I can find the horsehair worms that were observed here, 3 years ago or something, right? We can collaborate with other parasitologists, but also entomologists. A lot of people who study insects, they'll collect the insects and all of a sudden this worm comes out of it. They're not studying the worm, but they can contribute. citizen scientists, of any age can do this. Everyone now has iPhones. Little kids can do this. And it would be nice to have a global collaboration, kind of leveraging the power of the internet for something that's actually useful and edifying. So just want to thank, a lot of this research was funded by the Haddock's President Faculty Research Award. The Jesuit community here at Creighton has been very supportive. One of my students this year had a Claire Booth Luce scholarship. And a lot of students are just working for the fun of it. So just thanking all of these students, very extensive list going back many years. And with that, I'll take any questions that you might have.
Thank you. Thank you so much, John. Well, this was really a fantastic presentation, very scholarly presentation based on your own research area, your own field work and the lab work. I'm sure this really inspirational for the students from the point of view of encountering a research methodology over there, the passion and the commitment behind an authentic scientific research. Actually, we were taken into the amazing world of insects and parasites, how small they are, how beautiful they are, how mysterious they are, but an authentic scientist researches into or exploring the mystery of the being. That was the beauty of this presentation, and a lot of information, database information, results. This has been an experience, academic experience for the students who are participating in this. And I'm really personally fascinated by the final invitation for collaboration. I'm sure there is a lot to collaborate with parasitologists, global collaboration with entomologists. So a lot of opportunities. And for the students and the members of the faculty here, this is a wonderful opportunity to discuss with our resource person, Dr. John. And I'm sure John is all the more open to receive your questions. It could be questions, it could be your doubts or clarifications, your own comments, or anything that you would like to communicate regarding this topic. You are most welcome. You can also ask about the Creighton University's other areas of research. So students and the members of the faculty, you are most welcome. Over to the floor.
Thank you very much, Dr. John SHEA. It was a very amazing section because I am an entomologist. I am working in the field of entomology. So it was really a new approach that we are getting because in our country, in Kerala, we are using another approach. This is a very good approach and our students can follow this systematic approach. And I think they are getting more idea about how the research is going on based on the basic ideas of science, especially in taxonomic field. It is very interesting and we are also very enthusiastic to participate in that final collaboration that is told about that. I have a doubt about how we can use this nematode parasites as ecological indicators. Normally we are saying these parasites are actually they are parasites, no? Then how we are using this as an ecological indicator in the systems?
Right, yeah. So normally people associate parasites kind of with bad things, but some parasites, not all, but some parasites have these complicated life cycles and they involve multiple hosts. And the more well-studied example are these trematodes. And so trematodes, the adults are in some sort of vertebrate host, and then eggs get passed out into and end up in the water, and they hatch out into this larval stage. It will then find a snail. They'll infect the snail, develop inside the snail. Then they leave the snail and swim to a second host, right, and develop in there, or insist in that second host until the vertebrate definitive host consumes it, right? So you have three different hosts, plus you have, this stage where two stages that are swimming in the water. And so if the water's polluted, that stage may not do so well. If any of those hosts are absent, right, because they're usually that first intermediate snail host, it can't just be any snail. It's usually a very specific type of snail. So if the snail host isn't there, or the second host isn't there, or definitive host isn't there, then that life cycle is not going to continue. It will end. The parasite will go extinct in that local area. And so what's nice about the trematode system is that you can just collect the snail and find the larval stages of the trematodes. And if you find a diverse assemblage of those larval stages, that should indicate a healthy functioning ecosystem. And because not only does it indicate that, okay, the water's clean enough for the larval parasite to swim in the water, it also indicates that the hosts are also there, and the hosts are doing what they're supposed to do. The hosts are, the otter definitive host is eating, the tadpole that has the parasite cyst stage in them. So it's functioning. The ecosystem is functioning, and you see a diverse assemblage. So I'm kind of curious in doing it with nematomorphs, because they also have complicated life cycles. They also have that free swimming stage in them as well.
Thank you, sir. Thank you, sir.
Welcome.
I'm not a biologist. I have a general question. First of all, let me record my appreciation of your wonderful presentation that was intelligible even to an outsider, a non-biologist. I am a physicist, a theoretical physicist, but I would like to ask one question about behavior. From what I have gathered from your presentation is that your main focus is behavior of these parasites in different environments. And coming back to the first slide, where you described the behavior, the mating behavior of some parasite. I cannot recall the names, excuse me, but the variation in their behavior between male and male, male and female, and female and female is intriguing. Is there any sexual importance to this? Are some pheromones involved in promoting this kind of coming together? And I have noticed that you have pointed out that these ratios depend on the behavior, depends on the environment. In aquatic environment, the percentages are different. So, does it indicate the role of pheromones in promoting this behavior and in different environments?
Okay, yeah, thanks for that. Yeah, we are actively looking at the pheromones. So the data, the studies I first presented, we didn't do any pheromone studies or anything. So we just, we didn't know if there were pheromones. So and instead of like looking for something that may not exist, we just want to say, okay, if they're non-randomly encountering each other, then that suggests that there could be a pheromone. Whereas if they were randomly encountering each other, then we're not going to bother wasting our time looking for a pheromone because it's probably not there. So because we found some evidence that it was a non-random encounter, we decided, well, okay, now it's worth like going looking for the pheromone. So I didn't present this data, but I was working with a colleague in chemistry, and some students did some, analysis of like the hairworm cuticles, of looking at male and female hairworms, and also looking at the water that they were kind of living in, and seeing like, is there any difference between kind of the male female cuticles and water solutions that they're in. And we think we found it. So I ordered online this chemical, and it really arrived maybe a week or two ago. And I have a student that he's in the lab. he's going to come in, this summer and use this chemical and sort of, can he stimulate the male worms with this chemical, or the female worms, or sort of, does the chemical going to give any sort of elicit any behavioral difference in these worms? And if so, then that suggests that, yeah, we actually did isolate what is some kind of sex pheromone. That would be really cool. Thus far, the chemical is very hard to dissolve in water. We have to like stir it really hard and heat up the water. And then we're not convinced that it's really doing a whole lot, but it's early stages, so I don't want to say too much, but we are looking at pheromones.
It is very interesting. I hope you will proceed.
Thank you.
John, how is the summer school going on in Creighton right now? We all know that this is the pandemic time, and how is the situation there? Are the programs still online, or are there physical schools in format? Could you just give us some idea about that?
Yeah, so actually the past year we have been meeting face to face. We had to limit the number of students in the classroom. So they had to be spaced out, or we had larger classrooms, or some had to be online, and the other half had to be, in the classroom. So it was, for my labs had to be like split in half. I moved to a larger lecture room, but we've been meeting face to face, and everyone had to wear masks and everything. This past summer, a couple weeks into the summer, Creighton University dropped the mask mandate. They dropped pretty much looked at all the restrictions. So there are summer classes going on. Students are meeting face to face. It's about as normal as it was before. We're very fortunate to have the vaccines have been working, and the number of cases, at least in the university community, has been very, very low. Like, as far as I know, I don't think they stopped reporting it. Honestly, they don't even update it anymore. So I couldn't even tell you what the numbers were. But so yeah, we're looking forward to a fall semester that will be, just like it was before the pandemic. So.
Thank you, John.
A general question.
Yeah.
What is the reception of your students to online classes? Have you switched over to online classes completely, or certain percentage? And the outcome?
Yeah, so it kind of depends. Some courses, everything went online. Some were what we called hybrid. So half the students would come to class, the other half would be at home watching it on their computer. And then others were all face to face, but again, it was in a larger classroom. Everyone was spaced out, wearing masks. I would say, like, in general, the students did not do well online. As a general rule, there were some exceptions where students actually did better online, but the general consensus is that students did not like the online courses. They did not do well on them. They were distracted. It was easy to sort of, maybe skip the class, or just like watch the recording of the class, and then they would fall behind, and they didn't have the interaction with other students that kind of like motivates them, keeps them going. Yeah, in general, it was not a good situation, I think. For
same experience here also.
Yeah.
Everywhere.
I think so.
Were you following this pattern before the pandemic? Did you have hybrid classes?
No, we never had hybrid courses. We did have online courses. They were usually for students that were like professional students, or like, they finished their bachelor's and now they were returning and getting like another degree in something, and so they were more motivated, I think, to take those online classes. Yeah, but this is the first time there. I mean, I guess there were some good things about the online thing. So my labs had to be split. And so half the students would come to lab in the first half, and then the other half of the students would come for the second half of the course. So there were smaller labs, and the students got like more attention. And in some of the cases, like learning how to use a microscope and learning how to use oil immersion, it worked great because like they got so much attention, even though, you know, what would normally be a 3-hour lab is now an hour and a half. In that hour and a half, they got enough attention and they were able to like use the oil immersion, and I had no problems the rest of the semester with oil getting on the other lenses. Whereas in previous years, a lot of students were I don't know, they fell through the cracks or something. They didn't learn it the first day, and then everything got messed up on subsequent labs. So I will probably, at least for that lab, I'll probably have only half the students come in for that lab and split it up, because in some cases it did work better that way.
Okay, I think I would love listening to some of our students too. If you have some final questions, 1 or 2 can raise your questions. This need not be question 2. As I said, could be comments or suggestions. Would you like to ask something to John? Well,
what can you say about the scope of parasitology? This is from Irene David. Yeah, and I'm not exactly sure what you mean by scope, but so I study parasitologists, parasites. You know, is anything that is derives benefit at the expense of another organism, right? The host, right? So technically, you know, viruses are parasites if you consider them to be alive, but bacteria are considered to be, you know, some bacteria are parasitic, right? So the scope of parasitology could include viruses, bacteria, but also tapeworms and roundworms and these nematomorphs, and right? And so the scope can be very broad based on the definition, but in practice, the field of parasitology, right? The viruses, virologists study the viruses, the bacteriologists and microbiologists, they have the bacteria cornered, right? And then the wormy things, I just tell my students, the wormy things are what the parasitologists study. I mean, we do malaria, we get some protozoans in there as well, but yeah, and I guess I don't know if that answers your question, but in terms of the scope, it can be as broad as you want it to be, but in practice, it's usually the wormy things.